Good morning, everybody. Great to see you again this morning. A little bit warmer this morning. For those of you online, thanks for joining us again from near and far. We are into day 3. You're over halfway. Well done so far. Hopefully you're still enjoying it and the practice are going well. We have a great lineup again. I've talked to you about how the days fit together. It's going to be very similar today. We're kicking off with a few insight talks, this time giving you a bit more of a perspective into medical devices and componentry like that. So Insight 5 and 6 will cover those aspects. But what I'm going to do now is move straight to our sponsor video so we can keep to time, and I think, all going well, there you go. These are our speakers today. You'll meet them in just a moment. The thin films. I should really follow your slides, Bill, don't you think? The thin film tour is on today. If you'd like to see you heard about the coatings on Tuesday, how to coat materials in vacuum. We've got some amazing facilities here on campus. If you're interested in that area or just even a little bit curious, jump into that tour. We don't have a limit or you don't need to register or anything. Just rock up at 12:40 in the foyer, so after lunch, and you can jump onto that tour. So now I think it's the video from SciTech, one of our wonderful sponsors that make all this possible and make it free for you. As I always say, when you're famous and rich, go and buy lots of things from them to thank them. Here we go. Sorry, let's get rid of that. Let me just jump out of that. So a big thank you to SciTech for sponsoring the winter school. I should really turn off my phone. Apologies, everybody. Which scene do I go to? All on, Bill? Let's try that. So you will have seen the presentations, the right-hand side. And I should have done seamless transition. I'll take the hit for you, Stephen. Let's try that and that and back one. So what I was going to say before is that you will have seen the tables with the displays from the different sponsors. Please go and introduce yourself and see the technology there. I wandered over and had a look yesterday. It's probably going to cost some money because I saw something I liked. But there's some really amazing technology there. I was looking at 3D microscope and other things. So please go and chat to them, get to know them, it's great to have good relationships with the sponsors because they can help us a great deal in our technology innovations.
Can I just mention to everyone as well, Evident Olympus, our sponsors who are supposed to be here running the practical session. Claire's been sick, so some of you who have done the characterisation practical have met her online. Craig Noble, who is supposed to be here today and tomorrow, is also really sick. So Ting and Hong Shu, who helped us out earlier in the week, are going to be running those practical sessions. But I will be putting some goodies out on the Evident table at morning tea. So please, be kind to each other and share it around, but there'll be some goodies out there for you to help yourselves too, and they do apologise for not being able to be here in person. They've got a really awesome microscope that's sitting in a box that we have not been able to open, which we're hoping to share with you. So it's a bit of a bummer, but at least you've still had the opportunity to enjoy the DXC, I think. I can't remember what it is, but yeah, the DSX. Yeah, thank you. Sorry.
So, as you can see, great support, and we've had Claire pushing through online for a bit while she was unwell, and we're making the best of it as we can. So,
check your volume, please.
Check my volume.
Yeah, are you on? Yeah.
You can't hear me?
Yeah.
I'm on.
That's right. I've got some challenges here online.
I was probably hearing you because you're over there.
Oh, maybe it was me. Yeah, go on, you keep going.
All right, online. I hope you can hear me. We can hear me here. Excellent. All right, let's move to our first Insights speaker for the day. It's Stephen Blakeney. I mentioned before that ANFF-SA is a partnership between the University of South Australia and Flinders University. We work very closely together. We've got complementary expertise. We partner up on many, many different things. And one of the amazing things we've been able to achieve is to have Stephen, who actually moves between the two facilities. He's based at Flinders but spends time sitting in the ANFF team here as well at Mawson Lakes. And he's an expert in the medical devices field. He's come to speak to us today on that topic, but let me just introduce him as the National Innovations Manager for the Medical Device Partnering Program, MedTech Venture Partner for the Target Translation Research Accelerator, co-lead for the research translation at the Medical Device Research Institute, and a guest lecturer and mentor for the Innovation in Medical Devices topic at Flinders University. Stephen has worked on a wide variety of devices, and in his current role he supports medical device companies around Australia, speaking to at least 50 to 100 medtech startups per year and providing advice and guidance on how to commercialise their medical devices. As you can see, he's a real expert in this area, and it's a great privilege to have you join us today, Stephen. We're looking forward to hearing from you today. Let's make him welcome.
The first step is trying to get over this thing without falling over, so success so far. It's a risky question, but is anyone involved in the medical device space interested in it? Because I realise if no one puts their hand up that I should probably sit down again. Oh, good. All right, there's a few people. Yeah, so I actually studied mechanical engineering, thinking that I was going to get into Formula One aerodynamics. That was what sort of got me interested in engineering. And when I graduated, I had a choice between a job working on a deep brain stimulation piece of equipment, or I could go into defence stimulating missiles, and I went for the medical device option and never really didn't know too much about medical devices at the time. But it's been this huge opportunity, huge field that I've kind of really enjoyed getting to know more about. So I guess you never know where you'll end up. So even for the people who didn't put their hands up, maybe you'll move into medical devices, find a bit of a niche in there. So I work for the Medical Device Partnering Program. I'll explain a little bit more about what we do in a moment. But I guess previously I've worked in industry, worked for medical device consultancies, working on a whole range of different, sort of more on the hands-on engineering for different medical devices. Did work on that deep brain stimulation. So a bit of experience all across, from sort of early stage ideas all the way through to products that are on the market. When you work on a deep brain stimulation machine, it takes a long time to get to market. So that was quite a small chunk. But then some of those other projects, you sort of get to see them all the way through from start to finish. So I'll do a little bit. I don't want to talk too much about MDPP and what we do. I want to talk a little bit about medical devices more generally as well. But we'll start with a little bit about our program. So I can't remember the exact date, but we're almost on our 16th birthday at some time in July. So 16 years, fully supported by the South Australian government the whole time. We've had support through the federal government as well, and through the Victorian government. We had a Victorian office for a few years. And in that time, more than 130 projects, and we try and keep track of some of the startups that we work with. It's hard because over 16 years, people change and we can't always keep in touch with everyone. But in in 2023, you can see up there, we found our companies had raised at least $23 million of what we could find, possibly more that we don't know about. So the program it's sort of evolved a little bit over time, but the concept at its heart has always stayed the same. So successful applicants get 250 hours of technical expertise, and they also get a bit of a market assessment. So we do some due diligence to pick the projects that are successful. Part of that due diligence we like to capture and give back to companies, because they're all inevitably they're early stage companies and they're going to go on to try and do some capital raising, or they're going to apply for grants, and some of that due diligence that we do we like to share so that we're not all repeating ourselves. So it gives them something that they can then share on with other organisations afterwards. The 250 hours of technical expertise, often it's prototyping or proof of concept type work. We have occasionally we do sort of benchtop studies, but the idea is that we're independent third party, so, and we've got a really good reputation across the country, so a lot of credence to the work that we do. We don't keep any of the IP that we generate, so everything we develop, all of the technical files, everything we do, we hand over, and we also keep in touch with people afterwards, so if they have any questions, then we're happy to help them. But really the idea of the programme started from the idea that Professor Karen Reynolds at Flinders really wanted to get ideas out of people's heads and into reality, and get rid of all of the barriers that stop people from talking about their ideas. That people are worried about losing their IP, they're worried, especially talking to a university, they think you want their IP. Contracts can be quite hard, so we're really streamlined in the way we do our contracts, all the boring stuff behind the scenes, but makes a really big difference to how we can work with people. So the only thing we ask for the organisations that are successful through our application process is that they pay $5,000 co-contribution, and if there's costs of bits and like consumables and stuff, we pass that on to them as well. So the flowchart at the top is our application process. I won't go through too much. The one stage I'll mention is that we do have a workshop, which is where we get lots of different people with lots of different expertise together. So that can be manufacturers, clinicians, regulatory consultants, all sorts of different people. Sign a confidentiality agreement and they all come and sit together, which is a really rare opportunity. So companies often being close to their product often would struggle to get this sort of group together. We're very lucky with the background that we have that we're able to pull together some really good groups, pull on a lot of the clinical networks. Over the 16 years, we've met a lot of clinicians, a lot of very innovative clinicians, so we get to bring them together to give feedback and help to develop products. And then the other thing is we have an independent assessment panel that approves our projects. So we run about 6 to 8 projects per year, and we get about 50 to 100 applications per year, so we've got quite a lot of whittling down to do. Someone asked me what the disappointed, the people that we let down, whether they're disappointed, but luckily we're able to help most people. So for the ones that aren't successful, we have really good networks, so we're able to connect them in with, we'll either find something that they were weak on in their application. I normally blame our independent assessment panel and say, unfortunately, you're not going to be successful going through to our independent assessment panel, trying to give them some feedback on the types of areas. So sometimes they're really coming to us with a research problem, but approaching it as a startup. So we might connect them with researchers who are able to help them to prove sort of the underlying technology. That's in a case where it's sort of maybe analysing some sort of biomarker or something like that, where we need some evidence. We do get clinicians who come with something scribbled on a back of a paper towel, and that's fine if we can sort of understand that it's technically feasible. But there are situations where maybe the commercial viability hasn't been proven, so there's some programs that help to help people develop the commercial proposition, so we might send them to some of those, or we might connect them in with organisations that help to develop that. Sometimes they've got a few pieces of the puzzle. Might not have, they're strong on technology, they're not strong on commercial, and we can connect them in with groups that can sort of add that extra flavour. So no one ever comes back upset. We always manage to help everyone. And the South Australian government are very hot on the fact that when we talk to them, we talk about the projects we do, and they say it's not the projects you do, it's the fact that you help everyone with a medical device idea that comes to you is what's really important. And Craig mentioned the Targeted Translation Research Accelerator that I'm involved in. So we're partnered with MTP Connect on some big national grant programs. They're MRFF-funded programs, and we're the MedTech expert. The BTB program on the right's finished, but there's a couple that are still running. No funding available at the moment. We're just mentoring companies that were successful through the projects. But some of you may, TTRA has a couple of research centres connected to it as well. So one for diabetes, one for cardiovascular disease. So some of you may have come across that. If you want to know any more, I'm more than happy to have a chat at morning tea. I just thought I'd quickly include this. This probably more sort of from an internal process, but people just, I guess, to explain where the MDPP and ANFF fit. So we've been running this program that delivers the 250 hours of technical expertise, and my role and my staff are involved in that application process and figuring out what sort of projects we're going to do and trying to get approval for them. But really our engineers are all part of our medical device prototyping facility, which is part of ANFF-SA. So we've got some equipment, some prototyping equipment, but a lot of the expertise that we have is the engineers. So they've understand medical device processes, materials, all sorts of things. So really the people are what make us really good at what we do. This is a quick, I think it's a video of the lab. So we've got a few examples on there of some of the devices that we've worked on. I will do a couple of case studies in a moment. Got a good design award, and then some equipment. So we've got big and place machines and 3D printers. I mean, it just looks like a regular lab, but there's lots of really cool stuff in there. We've got some simulators for testing medical devices as well. So we do that early stage ideation proof of concept. What we'd like to do is a bit more of the later stage, but that's coming soon. But one of the problems we do see for medical device companies that we work with is it's a really long path to market. There's a lot of things to consider, and particularly the people come from either research or from a clinical background don't necessarily have the understanding of what it takes to get a product to market. Engineers tend to be a little better. They're more process oriented, and a lot of the taking that product through those last steps. So we had a inquiry come to us recently. It's a very simple, technically very simple device, but it's a class 2B, potentially class 3 device, which is almost as hard from a regulatory perspective as they get. So from a paperwork side, going to be really hard. From a technical side, really easy. And people don't necessarily see it that way because they figure easy technology, I'll get it through, no worries. So in terms of case studies, so this was a urologist who came to us with, he wanted to develop a urinary catheter. So Foley catheters, they sit in the bladder for 72 hours. One of the challenges they have is that male urethras have a prostate, which causes a bend in the urethra, which can make it quite difficult, particularly normally in first instance, a nurse will be the one to catheterize a patient. So not a trained urologist. And if you can sort of see at the bottom of that translucent image, there's a little cavity. So what can happen is as they try and push the urinary catheter through, they can actually cause some damage to the urethra, creates what's called a false passage. And then the catheter will tend to follow down the false passage. They do have some other tools that urologists will use. So they have a metal introducer that goes through the middle. They have a catheter that already has a pre-curved tip. Astonishingly, they don't have a catheter with a steerable tip. So Ian Middleton came to us with this idea. He'd already talked to a patent attorney. They always do. They should come and talk to us first, but they always talk to patent attorneys first. We helped to develop. So we had built a full-scale silicon mold, 2 different types of silicon to control the bend, and running an extra lumen through the middle with a cable attached at the end to a lever that you can use to manipulate the tip. Idea being, the nurse will put it through. So first attempt, put it through till they feel a bit of resistance. Then they can deflect the tip and they can follow it around. So hopefully means that urologists don't get called down in the first place. Very low-cost way of doing it as well. This was a Victorian company working on an alternative to CTG monitoring. So where CTG uses ultrasound, this uses more similar to ECG. So it's got 7 electrodes. They did a lot of research into electrode position and what they're able to have, an algorithm that basically separates out the mother's heart rate, the baby's heart rate, and you also want to measure intrauterine pressure with these devices as well. So, don't know, anyone who's had a baby or had a wife or partner that's had a baby will know that midwives spend a lot of time moving the CTG monitors. They're not very reliable. They've got to have a line of sight to the baby's heart, and babies tend to move around a lot and don't play ball very well. So the alarms keep going off all the time, and midwives are constantly moving them. So the idea is that not only during labour would this make it easier, but it also gives obstetricians the ability to send patients away and be able to monitor them and be able to delay potentially inducing or performing emergency C-sections, because they'll be able to monitor babies. Particularly the obstetrician that was the co-founder of this company, she was really keen, does a lot of work in sort of remote areas, so she was really keen that she could support patients that weren't close to the hospital, that she couldn't get in quickly, that she'd be able to monitor them remotely. And I think they've raised about $500,000. So we did a project with them about probably less than 12 months ago. They've raised $500,000, and then they got $50,000 the other day. So raising a bit of money, a bit more work to go, but they've been doing pretty well since they finished up with us. And this one he's actually a vet. He came up with an idea. It's a bag valve mask ventilation. People tend to, it's a stressful situation during CPR, and paramedics are often doing lots of things at the same time. You actually, depending on the patient's size, determines how much you squeeze the bag. And it's about every 8 seconds that you squeeze it. And often, people aren't, they've got lots of other things to do. It's hard to keep track of how often you're squeezing, how much you're squeezing. So he came up with, he had a Raspberry Pi and a very basic prototype, which fell apart when we opened the box. But he looked at using infrared and different Hall effects and lots of different types of method to measure across the bag. So I should mention that there are inline sensors, but the idea is that with an inline sensor, you have to take the bag apart, put it back together again. This is purely outside of the bag. And yeah, we ended up coming with a whole new, and again, he talked to a patent attorney before he came to us, and we came up with a whole new way of sensing, which was much cheaper and much better, gave us the best results of all of the different concepts. So he had to go back. Luckily, he could still change his original patent, but this is the problem when people talk to patent attorneys before they talk to us. So yeah, that one finished up about 6 months ago. Not heard from him recently, but yeah, fingers crossed. So I think we managed to do a lot for him in this 250 hours. So we tested it with a flow sensor as well and got some really good results. All right, so I just thought I'd finish with just some hacks and thoughts around medical devices. So the vision that we have is we want to make Australia the fastest place in the world to take a medical device idea to market. And to do that, we probably have to stop making lots of mistakes, because people spend a lot of money they don't need to spend, and we see a lot of the same mistakes over and over again. So part of that is that those steps I mentioned earlier that we'd like to be a bit more involved in, but I just thought I would share a few bits of advice that pop up from now and again. So just on the commercialization side, one thing I see a lot, and there was actually a particular company that popped up on my LinkedIn this morning that made me think I'll add this, because they have a platform technology, and when they came to us, they told us about their platform technology. The problem is that I think from a research perspective, platform technologies are really good, but from a commercialization perspective, if you tell me you've got a device that can cure every cancer, it's very hard to see how that's really going to work, and you really need to focus in on find an unmet clinical need and fix that first. Having a platform technology as a background is really good, because you can say, once we've cured this one, then we'll move on to other cancers. But jumping straight in to say you'll solve everything tends to mean you lose focus, you keep following wherever the money is, and it gets very confusing. Another thing that we see people do is just find a random report that they found online and say the size of the market is $7 billion, but it's not really related to their products. So we always say bottom-up sizing. So think about the number of procedures, think about the clinical indication, you build up from there. That's applicable not just to medical devices, but really to any market, is try and do a bit of modelling and try and work it out and explain it. Another thing we see is when we do give people feedback, The ones that take it really well and do take action and then come back, we automatically know that we're going to enjoy working with them. The ones that are very defensive are usually going to be very stubborn, and their technology isn't going to move forward, because you're going to talk to people who possibly know more than you, but at least have some different perspectives, and they might be wrong, but at least if you try it, you'll have a better idea of where you're going, and you'll start to pick up some really good feedback. Definitely good to have an advisory board, and knowing your role, everyone wants to be a CEO, I don't know why, but we get technology people who just assume they're going to be the CEO. I don't think they really want to be. I think they like the technology and they want to work on the technology, but at some point you've got to change, and you can be the CEO to start with, but there's always a good point where it's good to switch and let someone else do that. So probably everyone knows there's a bunch of regulators, FDA in America, in particular I'm going to mention now, but we have TGA here. I did touch on this earlier, but medical devices are classed from 1 to 3, slightly differently between Europe and the US, in that we have 2A and 2B. It's the same here in Australia as well, and the US don't, but overall pretty similar. But in vitro diagnostics are classed differently, and people always come to us and they'll say, my IVD is a class 2A, and we're like, that's not an IVD class. So it's good to know that there's different classes. People often keep prototyping and then to turn it into a product, but actually for medical devices, at some point you've got to start again. You've got to go through an ISO 13485 design process. That's something that people mess up a lot, and they get a long way down the track before they realise, and then suddenly they've got to try and retrofit their quality management system to fit their product, and it doesn't tend to work. So this is something that we're really keen to try and get people working on more quickly, is to understand the process. This is where it becomes engineering. The example earlier of the class 3 2B or class 3 device, which is almost all going to be documentation, very little technical development. So one of the methods you can use in the US to get a medical device approved is the 510K pathway, which is very useful for companies, because all you have to do is prove that you're substantially equivalent to another device. The really cool thing about this is that they put all of the information up on a database, so you can actually go on and you can search for 510K submissions. So I use this all the time, and people think I'm really smart because I give them lots of standards and lots of information, but it's actually all publicly available. So just on this one, so I went to the database and put ResMed in, just as an example, picked one of their products. This is the S10 Kira. Go down to the summary, which is where you find the treasure. And then on there, so they've done comparative studies against, in this case, they've used other ResMed products, which is fine, you can use whichever. Some of them they use competitor products, but this can be a really good way to find data that's comparative across products, because what you often find is that, and they always say, for the regulator you tell them that your product is identical to everyone else's, but for marketing purposes you say it's completely different. So when you're looking at marketing information, it can be quite hard to compare, whereas this is the engineering data, so you can get some really good information. And then a list of the standards that they've tested too. So even if it's not perfect, it's still a really good starting point to give you an idea of what you're going to have to look at. And for early stage companies that normally have no idea, this is really impressive when people come to us with this sort of information. The other thing I thought I'd just quickly mention is that medtech trials are very different to pharma trials, and everyone knows pharma trials, but giving someone a drug and seeing how they react is very different to putting an orthopedic implant into someone, or testing a pulse oximeter, or all the classes of medical devices are so different, the way they're used is so different, so it's really very different to drug trials. So people often oversimplify. So this is the official sort of 3 stages. But I guess the main thing I would just say is that, which people often overlook, is that once your product is approved by a regulator and it's on the market, you're still actually in a trial, so that's the post-market surveillance. So you're constantly looking for adverse events. They can report it to the regulator, and you've got to action them as they happen, because in the example of an orthopedic implant, you could do your hundreds of patients for your pivotal study, but there's things that appear 1 in a million, 1 in 10 million, and it's really hard to pick them up in a trial. So this is why with ISO 13485 and the systems that people have in place, it's all about capturing that data and actioning things. And that's everything. And I think I've just got enough time for questions.
Thanks very much, Stephen. Really interesting talk. Great insights there towards the end of the talk. There are already hands up, so go for it.
I assume your eligibility for this program is only with Australian entities?
Yes, it is, yeah.
Would you be open to at least like having a conversation?
Yeah, very happy to have a conversation, and even if it's connecting you in with people, you can also create an Australian entity, and then you're eligible. But yeah, very happy to have a chat. Yeah.
What does your team look like in terms of size and range of sort of expertise?
So we're a small team. We're fortunate being within a university and being part of the Medical Device Research Institute that sometimes we outsource projects and we've got lots of experts that we can pull on. But we have 2.5 engineers, and we're about to hire a third engineer, so we're 3.5. A couple of people in the innovations team, but relatively small team. Hopefully growing.
Mostly the sort of technical sports side, not the market regulatory side?
Yeah. So my role is split, so I'm involved in that grant program and a few other things. So we have another CTCM grant program, so we have someone involved in that. So we're more commercialisation focused. We have an innovations manager who's more commercialisation focused, so we can pull on some of those different expertise. But yeah, more on the engineering side.
You mentioned that some prospects seek a patent before they come to you for services. What happens in that then? And that you prefer that they come to you first. What happens in that process if they do seek a patent first?
Well, it's just for innovation. It can be tricky if they come to us with a patent, then we're sort of tied to trying to create something that fits their patent rather than just truly being innovative. So like with the example of the bag valve mask monitor, fortunately we were able to change it, but the technologies that he put in his patent just didn't work very well. So it's almost and actually there's probably two things. One is that we're sort of fixing to try and work with people within the envelope of what their patent says. The second thing is that normally you have 12 months for your provisional, maybe 18 months for your PCT, and then you're at national phase where you've got to pick all the countries you want to file in, and the fees go through the roof. So really it's 2.5 years from filing your provisional to huge fees, and often you're not getting any benefit from your patent in those early stages. So you're better off trying to use NDAs and keep things confidential as much as possible, because so many companies struggle as soon as they hit national phase because they spend all their time making payments.
So maybe a follow-up question, Stephen. A lot of people, when they come to ANFF, or probably the same for MDPP, they're worried that they're going to lose their IP. So
yeah.
What happens in that case?
Yeah, I always tell people when they tell us they're worried about, cause we'll never push anyone if we always sign confidentiality agreements anyway, but usually in that first discussion we haven't, and people sometimes will be a little bit nervous. I say, it's up to you, you're more than welcome to share whatever you're comfortable with. I'm like, we know how hard it is to make a medical device. We don't want to steal your idea. There's no reason for us to do it. And in most cases, people, we have our jobs, and developing a product, which I think I demonstrated all the way through to market, is really difficult. So most people want to help and to do that a little bit, but they don't really want to do the whole thing.
More questions, yes?
And thank you for the presentation. So have you developed any actual classes?
As in products on the market? Yeah, so we've got, there's a few examples. So we did a project for a company called Ostafix that was for so they were a company that made orthopedic products, so nails and screws. One of the things they needed to do was to find, so at the moment they use x-rays to find where you put the screw towards the knee. So you have a jig for the screws at the top of the leg, and then you have a nail, and then down at the bottom they need to put 2 screws in down the bottom, and they don't know where they are. So usually they spend about 40 minutes of the whole procedure. Orthopaedic surgeons are very expensive. Operating theatres are very expensive, and 40% of the procedure is trying to find that spot. So they wanted to come up with a new sensing technology that could address that, which we did. This was probably about 10 years ago. We're on version 4 now. They keep coming back for us to develop new versions. But they've sold that to 40 countries around the world. It's been really good for them, and it helps them to sell all their different screws and nails. And there's lots of examples of companies. I think the challenge, although we've been around for 16 years, a lot of the products, so we had a rehabilitation skate post-knee surgery that took them 10 years to get TGA approval. It shouldn't take that long, but it takes them a long time to get regulatory approval. So a lot of the products that we're working on, I've been with MBPP for the last 5 years, so a lot of those are still going through that regulatory process, which is frustrating. It takes a long time for products to get to market, but yeah, definitely some of the older ones have been successful. We did some work with Ferranova, who are based here as well. They've raised, they're not quite at market yet, but they've raised probably about $15-20 million. So they do secondary cancer detection, and they're going really well. Yeah. Oh, perfect. Oh, leading into it. Oh, I shouldn't try and explain it then.
Let's say that was intentional. So,
Yeah.
I think it was a good example in this talk of how much help you need in innovation. It's something to be aware of, that there is help around. So if you're looking at medical devices, there's people like Stephen who have a great amount of knowledge, who've been through the journey many times. And if you tap in and, as your tips suggested, listen to the advice and take the guidance, you can speed up the processes of regulation approval, of some of those development steps, getting clinical advice. And so it's something that I hope everyone will take home from this school, is that here in SA and around Australia, there are people who can help you accelerate your innovation without stealing your ideas. ANFF doesn't steal your idea, MDPP doesn't steal your idea. We're there to help you succeed, and so feel free to come and talk to us about your ideas. We'll do everything we can. Stephen, thank you very much for your wonderful talk. Here's a small gift for you, and hopefully a lot of applause from everybody. Thanks very much, Stephen. So, as Stephen beautifully segued for us.
Sorry, just 1 second. A question has just come through. What sort of support do they do as far as helping ventures move to ISO 13485?
485.
Yeah, 485. Thank you.
I have to talk to the microphone so I can hear.
Yeah, sorry.
Just have to climb over the barrier again. So we don't do much at the moment. That's sort of what we would like to do a little bit more of, which is also a really good. I'm pleased that you raised it. I think there isn't a huge amount. I guess get in touch and I'll explain a little bit more. There are a few organisations that can help. It sort of depends on the product. But yeah, sometimes there's companies that will take on some of the regulatory responsibility for you if that's an easier path to market. But often it's a bit of a painful path at the moment, but I'm more than happy to chat.
Okay. Excellent. Thanks again, Stephen. Thanks for the question from online. As Stephen mentioned, Ferranova is a company that's involved in this wonderful process that's complicated but important for our future health care. And we've invited Dr. Nicole Dmochowska. There's a lot of letters there, and I've been told to ignore most of them. So I got close. Who's a research fellow and biomedical research scientist here at UniSA. Nicole works closely with Adelaide-based Ferranova, which Stephen mentioned, to develop cancer imaging technologies. She was awarded a PhD in medicine at the University of Adelaide and then joined Professor Benjamin Thierry's bioengineering group here at UniSA's Future Industries Institute. And her research is focused on developing small molecule tumour targeting imaging contrast agents, including nanoparticles, aimed at improving the imaging of primary tumour lesions. Nicole is a key member of Ferranova's research team that is building on a first-generation ferrotrace to develop ferrotrace FAPI, designed to target the stoma of solid tumours, leading to improved delineation on MRI. And very soon we'll all understand what that means. And so please welcome Nicole to come and speak to us today.
Thanks, Craig. So hi, everyone. I'm just going to give a broad overview of sort of two nanoparticle streams that I predominantly work on. So I know there are a lot of PhD students here today. When I was a student, I was always interested in what sort of pathway someone took to get to where they are. So just a bit about me. I did a Bachelor of Science in Chemistry and Pharmacology at the University of Adelaide, then honours in Health Science and Pharmacology, focusing on gastrointestinal physiology and pharmacology and inflammatory bowel disease. Then, as Craig said, I did a PhD in Medicine just down the road on North Terrace, South Australian Health and Medical Research Institute, or the big cheese grater that you might have seen on North Terrace. And that was focusing on radiochemistry, medical imaging, particularly positron emission tomography and immunology. So I guess key message here is don't feel like your degrees sort of restrict your pathway. All your skills are transferable. Right, so what do? As Craig said, I develop nanoparticles to improve the visualization and treatment of tumours. And this is presented through two streams, so the iron oxide nanoparticles as an MRI contrast agent, and a sort of new stream of work we're doing at the moment is dye-loaded polymeric nanoparticles for fluorescence-guided surgery. So we'll start off with the iron oxide nanoparticles. So as Stephen and Craig introduced before, this project is in collaboration with Ferranova, which is a spin-off company formed by Benjamin Thierry and Dr. Aidan Cousins. This is the Ferranova team. We're actually based up on level 3 of this building. It's a lovely team to work with, and they're also hardworking and just really want to make an impact on improving cancer treatment, which is really lovely. And this is one of the clinical MRI scanners that we use to scan our samples, and at the moment we were scanning a mouse, so it was a little bit like disproportional to a normal human. But anyway. Right, so why is MRI mapping of tumours important for treatment guidance? So doctors use MRI mapping for surgical planning, radiation therapy planning, and chemotherapy assessment of treatment efficacy. So particularly for radiotherapy, it's used to delineate the tumour and the healthy tissues, because realistically you could eradicate a tumour by just hitting it really hard with radiation, but what restricts the dose that is given is the damage to the healthy tissues around it, which then causes a whole heap of side effects, and the quality of life just decreases enormously. So also the inter and intra-fractional changes. So what I mean by that is when someone's having radiotherapy, they often go once or twice a week, over a few weeks, and there are changes that occur between those fractions. So they might lose weight, their organs might move around, or even during that one fraction of that one treatment of radiotherapy where they're breathing or the gastrointestinal tract is moving around. So the MRI can be used to sort of readjust the treatment dose area. And there's sort of this new technology coming through where the MRI LINAC, and the LINAC is the radiotherapy machine, so you'll have an MRI machine integrated into the LINAC, so as the person's having the radiotherapy, the MRI is guiding the treatment actively. So typically MRI works well, but the current methods are prone to underestimating the true tumour volume, which can increase the risk of relapse. So we're working on ferrotrace FAPI, which we've termed FRN111. It's based on first generation ferrotrace, and this is a project in collaboration with Dr. Valentina Milanova. So these are iron oxide nanoparticles with a dual-length stabilising and targeting polymeric coating. We have a long targeting polymer which has a ligand, a small drug on the end to target fibroblast activation protein. We have a patent in place for this. So what we hope is that by targeting the tumour via this FAP, we'll improve the delineation of the tumour border and its volume, and also decreasing the inter-rater variability. So the radiologist or the person that's planning the radiotherapy, there'll be more coherence in defining that volume between raters. So this could also enable high dose or boost radiotherapy doses, which is, I guess, kind of trendy in medicine at the moment, where you'll hit certain regions of the tumour with a higher boost dose. And it also could, by hitting less healthy tissue, decrease side effects and potentially improve relapse-free survival time. So this is, I guess, Ferrotrace version 1, the core IP from Ferronova. So it's based on a core iron oxide magnetic particle with this dual-length polymeric system. So we've got the long targeting polymer. The T here is the targeting ligand, and then the short stabilization polymer. So the first version of Ferrotrace was or is intended for the identification of sentinel lymph nodes for the staging and treatment planning of various cancers. So for Ferrotrace, they use a mannose, which is a sugar as a targeting ligand, as it aids to help with retention in high-risk lymph nodes. And this is currently undergoing clinical trials for oral, colorectal, and upper gastrointestinal cancers. So the idea is that the iron oxide core causes a darkening or a hypo-intense region on T2 MRI, which makes the tumour lesion more obvious to the clinicians. So talking about the target for our MRI contrast agent, FAP. So FAP is a cell surface protein that's highly expressed in tumour-associated stromal cells, but rarely in healthy tissue. So the expression in the tumour stroma tends to be enriched on the vascular niches of the tumour, which is a lot sort of on the boundary of the tumour, the new sort of forming tumour as well, which makes it an ideal target for nanoparticulate molecular imaging agents, as it's notoriously difficult to penetrate tumours homogeneously with nanoparticles due to the it's just a complete sort of anatomical mess with heaps of leaky vasculature and quite difficult. So if we're targeting sort of the strobal or the outside components, we should have a better binding there. So FAP is what's really exciting, actually. FAP is expressed in more than 90% of epithelial tumours, such as prostate, oral, lung, colon, pancreas, ovaries, and brain tumours. So this excites us quite a bit because if we can, prove that this product works in one cancer, it potentially has application in other tumour types as well. And this study here was a study looking at FAP-targeted PET. So this is positron emission tomography, and you can see that in all these different types of cancers, the FAP PET has picked up all these different lesions, and it's the same agent that's being used across all these patients. So to me, that's amazing. You don't see that very often. So we first tested FRN-111 in a preclinical model of prostate cancer. So you've probably heard about prostate-specific membrane antigen. It's the current gold standard target for treatment and imaging. It works great most of the time, but there's a small proportion of patients that have low or heterogeneous expression of PSMA, so it's not suitable in those patients. So then we asked, can targeting FAP instead be beneficial? So here's our iron oxide nanoparticle, its core, short stabilizing polymer, and the 30% targeting polymer. So on this yellow bowl here would be our targeting ligand. So we've got the FAP or the gold standard PSMA targeting ligand. So here's just a bit of characterization of the FAP and PSMA nanoparticles. This image here is a TM image of the iron oxide nanoparticles. So you can see they roughly have a core size of about 20 nanometers, and then here's a DLS image of the hydrodynamic side, which is approximately 60 nanometers. So then before we start injecting into preclinical models or things like that, we have to show validity or in an in vitro model. So here are some cell binding assays in so C32 cells are FAP positive. And so we have control nanoparticles that have no targeting ligand, and our FAP nanoparticles showed a significant increase in these FAP positive cells relative to the control nanoparticles, which is great, that's what we want. And the same sort of thing happened with the PSMA nanoparticles, where we tested these in LNCAP cells, which are prostate cancer PSMA positive cell line as well. So that was good. So then you may be wondering why we're using this 70 to 30 ratio of the short and long. And this is because the protein corona makes it notoriously difficult for small ligands to bind in vivo, because when nanoparticles enter biological fluids, proteins and other biomolecules absorb onto their surface, forming a protein corona. There's nothing to do with coronavirus. People It's just basically if you imagine a particle with a cloud of proteins surrounding the particle floating around, it can inhibit the binding because those proteins obscure the targeting ligands. So this can significantly influence the biological identity of the nanoparticle, affecting its cellular uptake, biodistribution, and therapeutic efficacy. Can virtually render it just not usable. Right, so here's just a sort of rough schematic that I've drawn. So by using this sort of two-length polymeric system, we have shown as well that these long polymers, the protein corona somewhat forms on some of the shorter polymers, leaving these longer polymers available to bind, versus if we used 100% targeting, there's just one sort of level where the proteins would absorb onto, so potentially having less targeting ligands available to bind. So then we did a bit of work as well looking further into this inhibition of target binding by the protein corona. So here we've got these strips of paper, and this is called a dot blot assay. So this row here is nanoparticles without a protein corona, so just binding on their own. We've got 0% targeting ligand on this left side and 100% targeting ligand here. So the nanoparticles are brown. So with 100% with no protein corona, there is a nice, very clear binding. But then once we introduce the protein corona, you can see there's virtually nothing there. So there is an inhibition of binding being caused by the proteins in this mixture. And what's interesting as well is we tested using 6 targeting ligands per polymer on the particles versus 2 to 3. And more doesn't necessarily mean better, because it depends on the configuration of these ligands on the polymer. In fact, the 2 to 3 targeting groups worked better, probably because their configuration was less influenced by the protein corona. So we tested the FAP and PSMA nanoparticles in an orthotopic mouse model of prostate cancer. So what this means is we take an immunodeficient mouse and we inject human prostate cancer cells directly into the mouse prostate. You need a microscope for that one. It's quite fiddly. So it's kind of a humanized sort of tumour growing in a mouse. So after the tumour's grown for about 6 to 8 weeks, we intravenously administered the nanoparticles, and then 24 hours later, we performed MRI. And so what we're looking at here is a section of a whole body MRI of a mouse. So we're looking sort of around the lower abdomen and the legs here on either side. So this blue is indicating the bladder, and this red arrow is indicating the tumours in the prostate. So on this left side, we've got the no nanoparticle control, which shows the intrinsic sort of tumour appearance on MRI, and then our control nanoparticles that have no targeting ligand. And we do expect some nanoparticles just to accumulate in the tumour just through circulation, so there's a small increase contrast there. But then our gold standard PSMA targeting nanoparticles is a bit more black contrast. That's what we're after in this tumour. But then in the FAP nanoparticles, we're quite surprised to see that these were even darker than the gold standard PSMA, so we're quite excited by this. And then we sort of went to more so quantify the amount of contrast that was being created in these MRI images, and we assessed the proportion of black pixels in these MRI images. And you can see there's an increase in contrast from the control to the PSMA and FAP. But then what surprised us again was this increase between the FAP nanoparticles and the PSMA. So this figure, as well as graph, also illustrates the sort of pixel distribution throughout the MRI image. So the left side here is the dark pixels and the right is the bright, and that's just the proportion of. So you can see even between the FAP and the PSMA, the FAP has much more black and a drop in this sort of light region here. And here we have an ex vivo, so just the tumour that's been resected out and put into some sort of solution that's undergone MRI, and you can see this really nice dark sort of region there. So our MRI results were further confirmed by histology. So here we've got some Prussian blue microscopy images, and Prussian blue stains blue for non-heme iron. So iron oxide nanoparticles will stain blue. So part of this, we were interested in whether the nanoparticles accumulate in the normal prostate, because we don't want that. So here's our known nanoparticle control. There's nothing there. The control, there was nothing. PSMA, nothing. And then FAP, no nanoparticles either. Fantastic. So then this is the prostate tumour here. And then we can see the PSMA has a nice blue boundary. And then the FAP also has a very nice blue boundary of nanoparticles. So the particles are accumulating along the tumour border, but also in those sort of perivascular niches around the vessels as well. So a few years ago, there was a UniSA study that showed that almost every blood vessel in glioblastoma, which is a brain tumour, was FAP positive, and that the normal tissue surrounding these tumours lacked FAP expression. So this makes it an ideal target for molecular imaging or mapping of brain tumours using FAP MRI. And as you can see, there are some immunohistochemistry images here. The purple staining is indicating the FAP, and the green is indicating the vessel outline. And you can see there's quite nice purple staining throughout there. So then we decided to start off collaboration with Dr. Lisa Ebert. And so looking ahead now, FRN 101 is currently in preparation for GLP toxicity and pharmacokinetic studies to enable phase 1 first-in-human clinical trials in recurrent glioblastoma and hopefully 2025. Right, so now we're moving on to the dye-loaded polymeric nanoparticles for fluorescence-guided surgery. So the surgical removal of tumours. In the operating theatre, surgeons rely a lot on just the white light visualisation. Surprisingly, their sense of touch, they like to feel all the tissue and the tumour, and which is quite surprising to me. So previous imaging like their MRI or any CTs that they've had, the knowledge of the anatomy and surgical judgment to guide the tumour resection and determine the margins. However, some tumours can be difficult to see, particularly when they are invading into adjacent organs, and some tumours can have these really diffuse sort of boundaries just leading into adjacent tissues. So fluorescence-guided surgery can help to mitigate the issues with just looking and feeling. So it's a surgical technique utilising fluorescent dyes to help surgeons visualise tissues in real time during the surgery. So what this can help with is tumour detection and margin assessment, ensuring maximal removal of the tumour, identifying critical structures such as vasculature, so you don't want to damage or cut or affect that structure, so preserving its function, and then also facilitate minimally invasive procedures such as laparoscopic or robotic surgery, because during these procedures you've got reduced tactile sensation and visual input. So if you're looking onto a screen, it'd be great to have this sort of fluorescent guidance. And then hopefully reduce complications and side effects by taking less tissue that needs to be taken. So here is an image of some tumour lesions in tissue without the fluorescence, and you can see there are some nodules there, and then this is sort of what it looks like with a tumour-targeted fluorescent dye, so that's quite handy. So moving on to what we've been doing, the indocyanine green is a dye that is commonly used in surgery clinically now. It's approved. It has great optical properties, but also has some downfalls, such as a really short half-life, poor stability in vivo, and poor tumour specificity. So the good thing about ICG is that imaging setups are already commonly available and used in the operating theatre, so we would have a sort of easier transition into the clinic. So addressing an unmet need. So towards addressing the challenges associated with fluorescence-guided tumour surgery, we have developed an ICG nanoformulation which overcomes the various limitations of free ICG. And this was by encapsulating ICG in a polymeric PEG-PLGA nanoparticle. And PEG-PLGA is widely used in drug delivery, and it's considered to be non-toxic. So microfluidic, this is how we make these particles by microfluidic nanoprecipitation. So we've got this nice 3D printed microfluidic device. We've got two inlets, one for the organic phase with the ICG dye and the polymers, and then one for water. So then these two inlets merge and then mix via this herringbone channel, and eluted in water is the ICG-loaded polymeric nanoparticles. So what's the benefit of using microfluidics versus bulk synthesis? So we get enhanced control over particle size and uniformity through the precise control of mixing, flow rates and reagent concentrations, improved mixing efficiency due to the high surface to volume ratio, uniform nucleation and growth of nanoparticles. And for us, it's quite scalable and high throughput, so we can easily scale from 1 to 2 mils to 1 to 2 litres. And because of the enhanced control over the mixing, we've got improved reproducibility as well. So we're lucky enough to have a MeCraft 3D printer in the lab, so we print these devices in-house. They are made out of PDMS, which is great for this application as it's largely inert and compatible with the required solvents that we need, and it has a hydrophobic surface, which means that the particles won't interact and accumulate along the device, hopefully. So this is our typical setup in the lab. We've got 2 syringe pumps, which pump in, and then it's just eluted out. So just quickly, not surprisingly, we figured out that by adjusting the flow rates, we could customise the size of these nanoparticles. And also we wanted to test whether the dye was actually also being encapsulated versus being absorbed onto the surface of nanoparticles. So as the nanoparticle size increased, the fluorescence also scaled with the nanoparticle diameter at a power of 3, which indicated that it was being encapsulated rather than just being absorbed onto the surface, which will be scaling at a power of 2. So our nano encapsulation does not degrade the optical properties of the ICG. So you can see the blue lines are the ICG, nanoparticles below on par. And importantly, the nano formulation is colloidally stable over 2 weeks. So we measured the size over 2 weeks, and there was not really a change in the size over that period. But importantly, the nano formulation's fluorescence over this time outperforms the dye alone. So as you can see, the clinically available ICG dye crashes out at 2 days, and our nanoparticles had signal still at 14 days. So this is sort of one of our setups for our endoscopic surgeries. So we've got Dr. Lauren Kennedy and Dr. Aidan Cousins. And here we've got a lymph node on day 0 that's been injected with our ICG nanoparticles, and then there's also signal still appearing in day 10. So due to the superior optical stability properties, we're able to extend the effective window of use, permitting surgery for much longer time, which would not be possible with clinically used free ICG. So looking into the future for these particles, we're looking into targeting FAP as well for tumour delineation, but also using it as a blood pool visualisation. So I'll just quickly touch on Lutfun's work. So Lutfun's interested in vaginal drug delivery. So she's looking at sustained release of nanoparticles in an ex vivo model of the vaginal epithelium. So vaginal drug delivery is beneficial for a heap of conditions such as preeclampsia or preterm birth, but it faces a lot of physiological challenges like variations in pH, the microbiota or the mucus. So she was comparing whether a gel or a liquid polymeric nanoparticle formulation had a better sustained release. So thankfully pig anatomy is like humans in many cases, so we could scavenge pig tissue from Samri. But then how do we use this tissue? Nanoparticle application needs to be controlled. So she's developed these 3D printed wells and this tissue holder where we would pin the tissue across this tissue holder with these pins, attach these wells and adjust the height with these screws, and then put this onto a PDMS filled Petri dish where we could put media in, so that's what that looks like. So that allowed for a really high throughput testing of her nanoparticle formulations. And this is just a nice image, a confocal image of the gel nanoparticles at 6 hours of treatment in red, and the cells are in blue. So she did show that the gel formulation versus the liquid did show sustained release. So you can see in the pink here, the liquid formulation peaks at 20 minutes and decreases at 6 hours, and the gel is the opposite. It peaks at 6 hours versus the 20 minutes. And thank you.
So Nicole, one question I'll ask right up front is you're dealing with nanoparticles that are coded to be able to target certain cells, and in principle that's a relatively simple concept, right? But the complexity of delivering it into the market and making it a usable device, I'm just wondering if you can talk about the merits of a simple concept being able to transition into a that journey, I guess. Because a lot of people make things too complicated in the start.
Yes, exactly. Yep. So it is incredibly difficult. And I guess where to start with this, you could have a whole hour lecture on this, as Stephen probably knows as well. But I guess it's about making your particles reproducible, but also keeping in mind that you need to be able to scale this up. Because what we do in the lab is completely different to what some sort of a contract manufacturing company can do. So Farron Over's encountered a few of those things. Like even some of the ways we mix the particles aren't able to do that at a scaled up batch. But apart from that, the small drugs are not too bad. It's just trying to anticipate, like you need to be sure that the product that you take into your pharmacokinetic and tox studies is your product that you want to use later, because once you're there, you don't want to change because you spent a lot of money already. Yeah.
Any questions for Nicole from the floor? Yes.
I was just wondering, for your, did you use any cancer on chip devices or models for your in vitro studies?
No, we haven't, but we do have people in the group working on those sorts of devices as well. We've got some people growing like miniature brain organoids in collaboration with Professor Cedric Bardia over at SAMRI. But not for my work,
Yes.
I have a question regarding the MRI contrasting agents. Since they are made up of iron, after the imaging, can we use it to induce hyperthermia for treatment?
Into what, sorry?
Induce hyperthermia for treatment.
I think potentially, but that's not our application that we're looking into. I know that some people do use those particles for that sort of treatment, but that's not our intended. We're also actually looking at the potential of these particles to radiosensitize the tumour by increasing the oxidative stress in the tumour. Yeah.
Any more questions for Nicole? Is there anything online, Carl?
Nothing online.
No? Okay, if that's the case, do you have a gift for Nicole?
I actually forgot this time.
It's not just me, though. All right.
Great,
Thank you very much, Nicole, for your presentation. Oh, okay. No worries.
Sorry, Craig, I've just taken over.
That's all right. Okay, thank you again, Nicole. Thank you to both our speakers this morning. A bit of an insight into the journey that you embark on if you're interested in medical devices. So it is morning tea. Our two speakers will be around the place. Feel free to collar them and ask them your further questions. The bell just reminded me to invite you, when you're in your pracs, feel free to take photos and post and do your socials, whatever you want to do, selfies with friends. Just enjoy yourself with that. We're happy for you to do that. But for now, I think it's off to morning tea, and we'll see you back here for the lectures very soon. Should I read things? Okay, welcome back to the room. You might remember from day one, I showed you a screen with all the different unit operations for microengineering, and one of them was subtractive manufacturing, either using laser like we saw yesterday, but also with milling capabilities. And we have a micromilling expert amongst us, which is Mark Cherrill. He's an ANFF-SA microfabrication technologist here at Mawson Lakes. If you get the chance, if you tempt him enough, he loves showing through the lab and telling you all the amazing things that can be done in our precision engineering centre. So if you tempt him, he will show you. Mark joined ANFF-SA after working as a mechanical designer for engineering companies in Adelaide on projects including oil and gas, water purification, rail and naval structures. Prior to immigrating to Australia, Mark was working as a CNC machinist and programmer for the Honda racing team and Braun GP, where he completed a Bachelor of Science in Engineering Design. He also worked with the Mercedes-AMG Petronas team in research and development. So Formula One experience, I'd say royalty amongst us. Mark's main focus is on design and manufacture of microfluidic devices and many other things using the Kira CNC micromilling machine. That's a little bit old because we used to have just the Kira and Mark working in our ANFF facility. We now have something like $10 million of micromachining capabilities, including diamond machining, and we've got 5 people working in that laboratory now, so which Mark is leading. So, Mark, it's with great pleasure that I invite you to come and wow us with micromachining. Let's make Mark very welcome.
I think I was a lot happier when it was just me, though, Craig. A lot less hassle, a lot less pain and misery. So it's become a bit of a tradition that I start off my talk with introducing Mike, who a lot of you would have met. Dr. Jing Hong Pai gave a lovely presentation yesterday. And half of you now would have had a practical with him. There's a couple of things you would have instantly noticed about Mike. Is one, that he's got a very impressive knowledge of microfabrication, and he knows a lot more about micromachining than I do about wet and dry etching. The second thing you might not have noticed is he's actually got beautiful hair. And before COVID, we used to encourage you all to go around and have a look when he wasn't looking and maybe just give a little stroke. It's really thick, black and glossy. Mike puts this down to his diet. Obviously, some of it's hereditary. And we're all very envious because we're all losing ours and it's going grey. Anyway, Mike's beautiful hair made him the prime candidate for some sample harvesting. If I can just get this to work. So we took a few samples, and you can see my hair there. Actually, this is many years ago now, when it wasn't sort of running away from my face. But this made Mike the prime candidate for some sample harvesting, and because we've got lots of beautiful pieces of inspection equipment, we were able to measure the thickness. And I think we've got a prize today, actually, because I usually ask you to guess the thickness of Mike's hair, and I think Belinda's got a prize for you. So would anyone like to guess the thickness of Mike's hair? You can just shout out. Sorry? No, so it's under 100 microns?
15.
No?
Actually, I can arrange a prize.
Yeah, so there will be a prize
There will
if you
be a
get within
prize.
5 microns. You can just shout out.
30.
No.
50.
Not far off.
70.
No. No, I'm going to give up now, I don't think.
They want a prize.
All right, okay, keep going.
20.
No, a lot higher than 20.
88.
Oh, 80.
95.
80, we'll go with 80, so the lad in the blue hat's going to get a prize at the end. So it's actually 88 microns. And our jealousy was well founded.
Oh, hang on.
That's fine.
I've got 2 prizes.
Did someone say 88? Oh, they can both have a prize.
I've
You've
got 2
prizes.
prizes.
Poster yesterday.
I'm going to have to choose a different intro here. Obviously, our jealousy was well founded because we measured all our hair, and it was like mine was 56, and no one actually got anywhere close, which obviously proves our point through science. Anyway, it's quite interesting to think that we could take something that's 18 times smaller than the thickness of Mike's hair. We can spin it round at 160,000 RPM. So this is like 2,222 times a second that this is revolving. And we can actually use it to cut channels in polymer and some soft metals, which kind of emphasizes the point, really, that how can we even kind of make this tool, and you can see here on the spiral geometry on the end, that can withstand the pressures of cutting. So this is a 5 micron diameter cutting tool. It's actually got a few diameters on the end. It actually steps down 3 times. So we can only see this properly or nicely under a few of the microscopes we've got. So it's an absolute nightmare to cut with, and we're only really removing like half or a quarter of a micron of material at a time. Just try not to stress this too much. So it's actually amazing you can actually make something this small and be able to cut material with it. So we'll take a little bit of a step backwards. So we're predominantly milling as opposed to turning on a lathe. So we've got a milling tool, in this case a sort of macro size milling tool. So we put this into a spindle, we rotate it, and we move the material through it. And every time it goes round, it cuts a little bit of material away. A lot of this is due to the geometry and the rigidity of the machine. So if you were to see one of these in action, if I can get this to work. So this is from a tooling manufacturer. There are lots of tooling manufacturers out there. So this is what it would look like. This is, 12 millimeter cutting tool. And you can see all the little bits of steel, or swarf as we call it, coming off of the tool. And one thing you'll notice is that the swarf that's coming off is actually a different colour from the parent material. So would anyone like to guess why the material is changing colour? Temperature. Yep, and as we go through this, you'll see that they'll put a competitor's tool up there, and it'll change colour again, and it will start to become blue. It's often said that this kind of straw colour is the optimum colour for the material you're cutting. So it shows you're taking some heat out of the material, but it's not getting too hot and damaging the tool. So you can see the competitor tooling now, and the swarf's going all over the place to kind of emphasise their point. But it's turning blue now, so it's obviously getting very hot, and this will wear the tool faster. It's like Ben alluded to yesterday in his laser machining talk. They don't get tool wear. And we do, which causes us problems down the line. So if we skip on to the next one now. So this is what it looks like when we're micro machining. So this is 10 micron ball nose, which I'll go into a little bit later on. And this is under a microscope. So we can't actually see the tool. We're cutting PMMA material. You can see a little bit of oil and air being blasted at the tool. We can't use conventional coolants on this tool because it brings the large debris back around. But as it goes up, you can see the air's being worked so hard as it comes off the end of the tool bit, because we're spinning around at 160,000 RPM. But it's actually creating these little vortices. So I'll skip on to the... So if we take a step back into the geometry of the tool, so we typically use 2 different types of tooling. We've got ball noses, which have got a radius on the bottom, and we use end mills, which have got a flat bottom. And inside the geometry of these tools, it's designed to bring the material in, curl it up into a nice little ball, and then it exits the tool as quickly as possible. So we're not recutting these chips. And a lot of research and development's gone into the geometry of tools over the years, because obviously if you can remove more material quicker, then you're going to save a lot of money in manufacturing. So what this looks like under the microscope, I'm going to show you maybe a couple of clips. I've actually got a better one than this. This is under an SEM, so it's showing you how the material shears away from the workpiece. And I'll show another clip in a minute. But you can see that this tool, which is uncoated, so we're going to coatings, and you would have all heard Paul's lecture on thin film coatings. So you can see it actually starts to build up on the edge of the tool, and that starts to cut itself. So we're actually changing the geometry of the tool now, which is going to cause us some more problems, but it's quite therapeutic watching this. I might just come out and show you this other one if I can, with some technical help, maybe. How do I exit this thing, Craig? Can I come out and I've got a link on here that I want to show because it's got a really, really cool commentary. Next slide. So if you exit this, so I can go back onto the desktop. Yeah, I should just drop that down now. I'll share the screen. So if we click onto here. Share again.
Menulog is now delivering from 7-Eleven. With hundreds of locations across the country, the
Edge. If you listen to the commentary, yeah, if we play that,
They're going to have to
maximise it.
have to share it first.
Yeah.
Where is it? Okay. Hopefully.
Yeah, it's a little bit grainy, but you'll get the idea.
Yeah, I'm just hoping online.
Occasionally, pieces of this built-up edge will break loose and escape with the chip. But hundreds of times each second, they pass under the tool point, remaining with the workpiece. This is almost the sole cause of poor finish. So not one, but 3 crimes against good machining can be pinned on inadequate cutting fluids. The bonding of metal to metal wears out the tool face. Friction keeps the shear angle low, causing more deformation, and particles of built-up edge escape with the workpiece and spoil the finish. But now, watch what happens when a small amount of a certain fluid is introduced. There goes the shear angle up. The chip immediately...
So you actually see that when we add a little bit of lubrication...
Welcome to my YouTube channel.
The shear angle, the angle at which the material, the steel, shears away from its parent material actually changes. And it helps us improve the surface finish, because before you saw there's little bits going underneath the tool. And a lot of what we do, we're interested in the surface finish. So the lubrication of the process is so important. Yeah, we'll skip on to the next one. Thanks, Craig.
Keep going.
So here's a couple of the machines that we've got at the minute. We've got the Cherrill on the right-hand side, which was the machine we started with. We've had this for around 12 or 13 years now, and I think Craig was here when they purchased it, because they had to get special permission from the Japanese government to bring it out of Japan, because they saw it as intellectual property at the time. Most CNC machines are run on ball and lead screws. This actually runs on linear motors, so it runs on magnets and air pressure, basically. I put 1.5 litres of oil in every 2 years. This system that operates the Cherrill gives it this accuracy. So it's actually got a 10 nanometer positional accuracy, which is obviously very important if you've got a tool that's 18 times smaller than Mike's hair, because if you haven't got that accuracy, it's just going to break and you put any side load on it. So the positional accuracy of the machine is very important. It took me probably 5 years to truly understand how this machine behaves, because obviously it expands and contracts with temperature in the room, if there's vibration in the floor, and quite often you'd set a long program running on a Friday and you'd come back on a Monday expecting it to have run 50 hours, and the tool would have broke 10 minutes after you left, and you've got no idea why the tool broke. You can't see the tool. So you've got to kind of delve down. So there's a lot of pain and misery involved with the micro machining that we do. We've got another piece of equipment here which we bought to make these small assemblies for DSTG. So it's on loan from the Defence Science Technology Group, and you've all met Igor, and there's a few other people I think in here from DSTG today. And this thermally compensates itself, so the spindles and things, they're characterized so it knows that whatever the room temperature is or the machine temperature is, it's automatically compensating. So if the room temperature goes up, it'll actually put an offset from the tool to whatever it's machining. It'll actually compensate back up to try and keep that stable position. Unfortunately, it's still on lead and ball screws, so we haven't quite got that accuracy, but we can still achieve that kind of micron finish. So I've told you some of these bits and pieces already. The Cherrill is a 3-axis machine. It's got a relatively small machining envelope. Obviously, it's trying to control that accuracy. Another interesting thing is we've actually got 2 spindles. So we've got an air spindle with ceramic bearings, and this is the one we use on tools under 50 microns in diameter, because we need that peripheral speed to cut. And then we've got an electric spindle which goes up to 50,000 RPM, which we'll do most of our cutting with. In industry, there's probably thousands of CNC machines in Australia. Most of them will only go up to around 10 or 20,000 RPM. So we're really lucky to have this high RPM and the positional accuracy that allows us to control exactly where the tool tip is and what it's cutting. So we've got a number of different tools here. We've got a drill in the middle that you can buy from Bunnings and you'll all have at home, or most of you anyway. And these are all end mills. All the rest of them are end mills, but you'll see they've got very different geometry. So the one highlighted by the green box, we've got a relatively slow helix. It's only got 2 floats. It's really sharp geometry. So this is for plastics and aluminium, because we can cut a lot more aluminium or plastic with every pass than we can steel. So it's got relatively open geometry, so all of these chips can exit the tool as quickly as possible. If we look up here at the one highlighted by the blue box, you can see it's a very different colour. So these are all sub-micron grain size carbide end mills. And this will go back again to Paul's talk the other day, because this has got a thin film coating on. So we've actually giving the tool different properties now. And they're even going to ceramic coatings now, where the tool doesn't even work properly until it gets up to 900 degrees. So this allows us, some of these coatings allows us to keep the tool sharper for longer, and it's not affected by the build-up of heat. You'll also see that the geometry on the tip's slightly rounded, because if we had sharp geometry and we were trying to cut stainless steel, it would just chip instantly. So we've increased the strength of the tool by putting a radius on there. So we'll have a little look at our production process, and I was talking to a couple of the people in here before we started. When I first started, students had come up, or industry, and they'd come up and you'd draw something out on a piece of paper, and you'd have these really bad sketches that you'd struggle to kind of interpret, and we'd sit down and we'd turn them into a 3D model in one of these software packages you can see here. But I think now, with 3D printing, you've already gone through these stages. You've already modelled it up in CAD. You've already made all these iterations to get you to a design now where you want to either show you can scale it up and make a mould, or you just want a really high accuracy piece to prove that whatever you're trying to prove will work. So once we got the CAD model, we take it through. We use SolidCam, this CAD-CAM software. So we turn in this program, we'll turn your CAD model basically into a series of G codes. And when 3D printers first came out, you used to play around with these G codes. Now it's all hidden behind the doors, and it just kind of does it. But it's exactly the same. Instead we're removing material, and wherever Daniel's going with his 3D printer, he's adding material. Once we've got the G code, we'll put it into the CNC machine and we'll make our part. In this case, it was a spiral separation mould to extract tumour cells from your blood. And as we go through this process, we'll be constantly inspecting it. This was a Vico optical profilometer, so we can inspect it to within a micron. And the inspection really is the thing we couldn't live without, because if we don't know what we're making, we haven't got a chance of applying offsets or knowing where we are. So it's a really important factor in our process. A few of the CAD models we did many years ago. Number one was a PDMS mould. So we did lots of different designs, spirals, pyramids, various other features in there, like bars and things like that, and wells. And they cast PDMS in here, and then they coated it with silver, and they tested the whether it increased the reflection. I think they were for headlights or something like that. Number two was a blood sampling device where you could do point of care at home. So you'd take your blood, you'd put it in this thing at the end, and it'd come down here, and these ceramic cards would take a reading. That was for a startup company in Silicon Valley. We've got another spiral separation device. Some embossing tools. So this was to put this helix into coal, and I think, who was it, Michael Bredmore spoke about coal yesterday. There was another lecture on it where they were flowing something over there to do with fracking, and I think it might have been part of the same project as that. We made this assembly here, so we've got a cam on the bottom, and this was to turn glass tubing around in a sputter coating machine, so they could apply a thin film coating to these parts right at the end. So we had to hold something that was very, very thin, fragile glass, and actually put a thin film coating on it. And number 6 was quite interesting, because a researcher came to us, and he was having to buy these disposable devices from Denmark, I think, and there was long lead time. So we actually made him a device that was reusable. So there's many different reasons why people come to us. And we're able to use CAD on this, or use the benefits of CAD on this, to work out even simple things like, can somebody's finger fit under one of these microfluidic tubing connectors? Obviously, it's a parametrically driven CAD software package. We can work out the centre of gravity, the mass. We can actually put many parts together in an assembly to check they fit. And here we've got a microfluidic device, and you can see here in red it actually shows you where the part's not going to physically fit together. So we can take out a lot of the problems, so that we don't waste time manufacturing, and then you have to go back to your workshop again and ask them to make it again. And obviously they'll get pretty grumpy with you if you know anything about university workshops. Usually from there, after we've designed a device for someone, or we've gone through this process with someone, we'll create an engineering drawing. And this is really the basis for everything, because we're laying out exactly what we're going to do, what it's going to be made from, what tolerances are going to be involved. And this is the specification that we then work to. So it's a fantastic tool for getting all of those little intricate details down. Once we've got our design, we'll take it into our CAD software. Here we'll select surfaces that we want to machine, or maybe a contour. For this device I had something like 72 programs, because they wanted like a micron accuracy. So it's a bit of a labour of love. Each one of those pockets, you might have these nice beautiful helical entries. If you've got a very small tool and you just go straight down into the material, quite often you'd break it or you'd damage the tool. So sometimes we need to enter the material very gently to keep the tool in good condition. And then the beauty of this is it allows you to simulate where the tool's going to go. So if you've got an orange line going off somewhere into the middle of nowhere, you know it's probably not good and you're going to crash the machine. And then we've got some rapid moves in between the programs. So it allows us to take out some of the problems we might encounter by seeing where the tool's actually going to go. I've got some pyramids here, part of the same project, and this will actually use a ball nose, and we'll actually go around here to create the surfaces around each one of these pyramids. Obviously we're doing this in series. The wet and dry etching, they're doing it in parallel, so this process can take a lot longer, but we can do a lot more different materials. So if we've got, say, a flat geometry on a thin substrate, I'd often send it to Ben at Macquarie for laser micromachining if it's just 2D, because he's going to do it a lot faster. He can just hold it down under vacuum. He doesn't put pressure on it like we do, so he can hold it a little bit more gently, and he'll manufacture it a lot faster. So when we talk about ball noses and end mills, these two images at the top, it shows you, so we'll take one of these yellow layers off every cut. So we'll take a series of cuts and we'll leave this little red zone on the end for a finished pass. So we'll run this at different cutting parameters, just trying to get that really nice finish and that accuracy on the tool. So these end mills, because they've got big open geometry at the bottom, they allow us to remove a lot more material a lot quicker. The ball noses, however, allow us to traverse these surfaces and get really nice surface finishes on truly three-dimensional geometry, which Ben alluded to is a little bit more difficult with his older setups with laser machining or things like etching, when it's a lot more suited to the sort of 2.5D, we call it, so more channels and slots and things. There's obviously lots of equations to this. As it's a sort of academic talk, I feel we need to put lots of equations in, but a lot of the time we sort of, after many years of pain and misery, you kind of go with your gut and you know what works and what doesn't. The difference is up here in this image. If you go with the wrong cutting parameters, you'll end up something like this, where you've got molten aluminium around your tool, and then you have to hide it from your manager and pretend you weren't silly. So it does matter. I mean, all these cutting parameters, we're running at 640 millimeters a minute at 160,000 RPM. Every time one of the teeth of the tool comes around, we're only removing 2 microns at a time, but because we can spin it so fast, we can actually move faster across the surface. when we've done all our CAM programs, this is what it'll come out with. And this, like I said before, it'll look a lot like the old style 3D printers. I don't know, do you still see this when you... Yeah, so this is all hidden now. But you used to be able to play around with this for better or worse, I suppose. That's probably why it's hidden now, actually. Because you can manually go in and change some of these parameters. And some of these are machine functions, but a lot of the time it's just X, Y, Z positions, and then a feed rate maybe. But you can see the level we've gone to on here, where we're going down to that sort of that 10 nanometers on here, whether it's truly at that position. Just like I said, the machine's 10 nanometers, and we're trying to achieve a micron sort of accuracy on the parts we make, which I'll go into in a second. So obviously after we've gone through machining, there's some of the devices that we'd make. So we make either very small things. You can see Mike's hair here tied through a part. And this actually helped us not to lose the part in the end. So we took a few more samples from Mike. And we actually just tied them in knots so you could see it, because these parts were going down to, I think this one might have been 0.5 millimeter in diameter. But we went down a bit smaller with 50 micron holes. And obviously even my hair wouldn't fit through that one. We've got an injection moulding part here, and these holes go down to 25 microns diameter, and this was to see if we could mould pillars. This surface, you'll notice, is a lot nicer surface finish than some of these other parts, and we went for a stage and we're actually polishing this surface. We can get down to maybe 30 or 40 nanometers polishing aluminium with a series of diamond solutions, and you get down to like a mole skin. But obviously it's only as good as how accurate your hand's going to run across, and you can, if you tilt it a little bit, you can obviously, you can put nice radii and things on the edges. So it's not a proven process every time. After that, we went to a different type of tooling, a monocrystal diamond tooling, which allowed us to and these are grown in a lab. They're single crystal diamonds, and obviously being a lot sharper and harder than the carbide we were using before, it allows us to negate some of the tool wear problems we have. And because it's so sharp, we get a really nice, almost optical surface finish. So we made this as a test part for after we moved the Kira to a different location. And we had 100 nanometer steps. So the edge of this part, which is about this size, is actually 3.4 microns lower than the middle. But you can see if you come and have a look at our facility, you can see where I get to the corners and I slow down the cutting parameters, and the pressure's there for a bit longer. It actually goes deeper. So there's lots of other things to consider. We've got a small assembly we've made here on a 5-cent coin. Like the slots in here will be 0.1. And these polymer O-rings or seals that we made, we couldn't physically make them without all these burrs on the edges. So we sent these to Bennett Macquarie to laser machine, and he got great results. We've got our blood sampling device, a pyramid electrode up here. So all of the tops of all of these were within a micron in height. And then we've got the actual part on here is right on the end. So we've got a concave parabolic mirror in the end. And some of our equipment, the Wilhelmin that I showed you before, we can actually hold this end part, reorientate it, and machine features in the back to accept optical fibres. So it's got a high level of fidelity. So obviously you have this problem as why doesn't the part look like my CAD model? So this is the world of pain and misery that we delve into, and this is why I'm losing my hair and it's all grey. Because there's lots of problem solving involved. And I've got a couple of examples here. The first one is the material. When it's made, there's often stress put into this material. So if we take a small layer off each side of this polymer, for instance, it'll start to distort and de-stress. So we might have to take the 2 red layers off the top and the bottom, and then take the other layers off just so we can de-stress it and hold it in a way where we're not putting stress onto it. Because even holding under vacuum, you're still putting stress on this part and it's still moving, and when you release it, it's going to flex back in a different orientation. And this doesn't usually matter for general machining in industry, but these small effect, when you're making really small features or small parts, it makes a big difference. We've got an injection moulded part here, and you can see at the end it's gone 0.35 millimetres in the middle to the end where it's bowed, because obviously this has been generated by melting polymer, putting it into a mould and coming out. If we haven't quite got those parameters right, then there's a lot of stress in that, and as soon as it comes out of mould, it starts to distort. We also have, when you're cutting and you've got a cutting tool in a large machine, and the machine might weigh anything up to 8 or 12 tons, you've got one piece of machine moving against another piece of machine, there's material and there's a tool there. Something often has to give. And if you've got a small, thin tool, or even the large half-inch ones, they'll still flex away a little bit from the material, and the ends might start to wear a little bit. So on this 75 micron high feature that we've got here, you can see the red outline. There's a 5 micron difference between the top and the bottom. So we've got a 2.5 micron wall taper, which is natural, a little bit like the ablation that Ben talked about yesterday, where the laser sort of induces this angle. We see it on a smaller scale here, but it's still a problem. We get other problems, like these two images here. That was the same CAM program, sorry. It's just on the top one, the tool was worn. So this little bit of wear on the tool edge caused it to look like this under the scan. It obviously took the edge off, so it wasn't sharp, so we had this radii in the bottom. When we reran it with a new tool, we got this nice clean geometry. So we often have to rerun parts, and we'll be inspecting them in the process to check that what we've made it is correct before we take it off the machine and send it to the customer. 3 minutes. All right, okay. Obviously, the effects of heat are massive because the spindles expand and contract. This is the same part, machined with the same tool. We ran it around this brown area first. It went off, it did some other features, came back around, and by the time it came back around, the spindle spinning at 50,000 RPM had caused it to expand by 6 microns. So it's the same tool, but we've now cut a groove in our part. So we often have to let it expand overnight, or it might take me a whole day just to set a tool, just so the machine's stable. With the newer machines, it does a little bit of this work for you, but then you start to fight this thermal compensation. And here we've got the same CAD model, same features, but we just put a 50 micron tolerance on these little parts. So the tool stayed within this 50 micron band, and you get this truncated feature. If we put a 1 micron tolerance, it stays within 1 micron of the diameter. So there's lots of different things we can put in to get better quality results, but it all depends on what the customer wants. So we're trying to achieve 1 micron accuracy, but all of these things come into play. So there's lots of cutting and measuring to know where we're going, and offsets applied, but there's lots of things to consider and try and understand. So in general industry, they'll use verniers and micrometers. We use a lot of laser confocals, optical profilometers, or for the really small parts, I had Mike's hair tied through the middle, we might do scans on it so we can look inside it. We've got a micro and a nano XCT machine, so we can look inside the part to see if all of the features are correct before we send it off. So I've talked a little bit about surface roughness and the monocrystal diamond tools, and this is the difference between them really. We've got 100 nanometers, which is actually pretty good, because in general industry, the general tolerance for surface roughness would be 1600 nanometers. So it's still very good, but it looks rough. Here we've used a monocrystal diamond tool to make this mold, and you can see the difference in the brightness of the material because of the sharpness and the hardness of the diamond. And when we talk about surface roughness, there's a few things to consider. So we have form, waviness, and then the roughness itself. So here we've got a form. We've got a convex surface. We've got a waviness, which is a machine-induced function, where you might have the bearings might slightly be out, or there's vibration when you're machining, so you get this sort of rhythmic finish that comes through the machine. And then you've actually got the surface finish itself. And this is the difference. You've got this beautiful lawn, and all we're seeing here, the grass is the same length, but we're just running up and down in different directions. So we're getting this very visual effect. So at the minute, we're down here with surface roughness around 20 nanometers. We've got less than that with the monocrystal diamond tools, accuracy of feature around 1 micron. So we recently moved to a new facility, and we'd built this home with all of those things in mind, like the temperature and the stability of the floor. So we built this home for the Kira, so we can optimize its efficiency, and we don't have to fight the room temperature and things like that. So it keeps the room within 0.2 of a degree during the day, and we're able to map that. We've got about 15 sensors in there now. So we built this environment, and we had space, and then our friends at DSTG came along because they'd had some equipment that needs this environment, and a lot of the times that people have got this equipment, but they haven't got the environment that it needs to live in. So since they've come on board with some ultra precision machining equipment, which I'll show you in a second, the surfaces can go down to 0.5 nanometer. And all your mobile phone, your camera lenses, the molds for those would have been made on this machine. So the surface roughness of that is going down to less than a nanometer. So that's like the size of a large atom, basically. On these machines, they make like the standards of measurements for the Kelvin, which they might make within 1 nanometer. Cranfield University have done some really interesting stuff online that you can see on YouTube. So now we've gone into this whole other world. So this is one of the pieces of equipment they brought in. So now we're talking accuracy of 1,000 times more, and it's actually got an 8 picometer resolution. So you're achieving these true optical surfaces. And luckily we're able to get someone with a lot of experience in this area because it's a whole other world when you go into optics. It's not surface roughness in nanometers, it's fringes, and I'm still asking him what's a fringe, and he's like, it's half a wave or whatever, and I'm like, what's a wave? So it's a completely different language that we've got to learn now, and I'm not sure if I'm too old for that. So Brendan, who's here, he actually made the lenses for this satellite that went up when he was at BAE, and Colin Hall put the thin film coatings on for it. This opens up other opportunities. So a lot of these mirrors and lenses, they've got thin film coating. They've been machined with a diamond. Some of them are glass optics, which will be polished. You've got things like CERN waveguides, which need to be machined with a diamond at a certain temperature, and it needs to be measured at that temperature because they take into account the thermal expansion. So all the dimensions on that will be within a micron because they might put 20,000 of those together. So if they're all at the top of the tolerance, the whole assembly will be 20 millimeters longer at the end. So some of these specify you have to machine with that. And this is the sort of surface finishes you're getting. Some of the materials you can machine. Obviously, it can't traditionally machine carbon steels because of the carbon on carbon will actually form a chemical reaction and wear the diamond very quickly. To get around this, DSTG have actually brought this ultrasonic. So this is reciprocating the tool at 104 hertz, which reduces the tool wear and allows you to machine some of the steels and other materials you can see here without wearing the tool. Obviously, inspection's even more important now, and the inspection equipment we've got is just not accurate enough. So they've brought a DUI, which just inspects for profile, so it doesn't do surface roughness or anything like that. $2 million, it just tells you what that curvature is. And we've got a white light interferometer as eco to measure the surface roughness. So Brendan will often inspect parts, and it'll look something like this. And you can actually see where, if you're holding it on 3 points like this, you can actually see where gravity is making it sag on the other parts. So they've got an optical flat, it's about this thick glass, as accurate as you can get, parallelism. They put it on these 3 points and they can see where gravity is making it sag in between. So it's quite scary, and he might be putting it on different things to try and negate this. Temperature and vibration are still a massive issue. And he shouts at us a lot for talking over the parts because the biological material in our when we speak, a little bit of saliva will come out, and if it lands on the part, it will start to degrade the surface. So we've gone into a whole other realm now of pain and misery, and then I'll leave you with that. Did I make up the 2 minutes?
Thank you very much, Mark. Fascinating stuff. Other questions for Mark? Yes.
We
we buy them, and we buy them from all over the world. This one's from PMT, and they make the 5 micron diameter ones, and they'll make some weird and wonderful ones for us, especially. A lot of the tools they make are for sort of PCBs and things like that, and softer materials. They've got very sharp geometry, but we get a lot from Japan and all over the place. As in physically in the workshop, or? Well, often you can't see the end of them, and when I first started, it was like just try not to get your hand anywhere near the end, because you just embed yourself with the whole cutting tool, and then you've got to order another one on a 4-week lead time from the US and try and explain why it's broken. So we do a lot of inspection on the tools before and after we cut, and we have to take them out of, like I'll know with a 50 micron tool I can cut for 60 meters at a 5 micron depth of cut. So we had to do a lot of work around sort of babying the tools to the end, just trying so they survive at the end of the program and not breaking them. So a lot of the parameters we'll use will be a bit slower than you should.
Any more questions?
Oh, there's prizes.
Oh, prizes.
It's a bag.
There you go. Mark,
Oh,
you promised it.
two. I did promise it that you've got a limited edition ANFF bag.
Well done.
Where's that? Oh, excellent. Oh, something's closed.
So just while we're setting up the next one, Mark, I'll ask you another question. We talked about your career being quite an unusual pathway to end up at a university. What can you tell us about, maybe just say something about your journey of how you ended up doing this ultra-fine machining in ANFF? Come around here.
I was saying earlier, I didn't actually know it existed until I took a job with a 6-month contract here, so I was either extremely desperate or I had the hindsight to see that this could be an amazing opportunity. But it's something very different, and it's nice to push yourself. Although it is pain and misery a lot of the time, it's nice to have that sense of achievement. You're not just making 100 of something in a workshop somewhere. But I was fortunate enough because I did an apprenticeship as well, and I did a lot of training part-time. So I spent seven years like doing night school and going up, and I did my degree night school, and my company paid for it, luckily. Just trying to get all of those bits of paper that you need to open that next door, and then just sort of, I suppose, going for it and trying to open that next door. I got to Australia, and I applied at 25, 30 companies. I got 2 phone calls, and one said, can you use hide-and-behind controller? And I said no, and I never heard back. And then someone took a chance on me to do some mechanical design. So it doesn't guarantee you're going to get that, so you've got to have a very thick skin and just keep trying, I think, is the message.
I think one of the things that has impressed me with Mark is when he joined us, we had, as I said, this one tool which was very unique in Australia, and it was not going to do any of the things that you could see on the screen today without one also excellent operator. And it highlights the story that we often tell, which is that to have the equipment is one thing, but to have the people and the equipment at the same time is another. And so at ANFF, we try and strive at having a fantastic team and assembling that team to be able to deliver what you need. And of course, all the things that Mark's just shown you are not things where you'll be able to walk up and use the diamond machining capability yourself or do some ultra-fine micro milling with these end mills. But you can tap into the expertise of the team, and they've already done the hard work. They've inspected the tools. They've worked out how far they can, 60 metres? Yeah. They worked out how far they can machine with those tiny pieces so that you don't have to do that. And so feel free. You don't have to be a specialist to be able to tap in and use the capabilities that we've got here.
I'm just downloading it, sorry. Somebody closed the file.
I thought I'm padding beautifully.
You're padding
but you don't have a USB, do you? Oh, okay. We're just going to USB it. So we're going to go very quickly, slowly from subtractive manufacturing to additive manufacturing. Most of you will have heard of 3D printing, and one of the things that's happening at the moment is people are very excited about 3D printing because it is a very exciting technique, But in fact, some of the capabilities that Mark's talked about, people came to us with 3D printed parts asking us to clean them up so that we could get this precision we wanted, they wanted. And we, in fact, switched around and said, actually, we can machine it from scratch. And so it's a good example of making sure you're using the right tools, the right capabilities for what innovation you're trying to achieve. Oh, good, we're on. But it's something that we try and offer you. If you're looking at some innovations, speak to the team, because they'll be able to steer you, whether it's to nano 3D printing, micro 3D printing, different materials, different milling capabilities, photolithography, bonding, all the things you've seen this week. You'll be able to get to the one that you need much more quickly. So, there we go. You didn't even notice we had a technical problem then, did you? So I want to introduce Daniel Williams to us. Daniel you will have met probably Daniel. Those who have done the 3D printing practice certainly have, and if you haven't, you will. Excuse me. Daniel's here at ANFF-SA as well in the Industry 4 test lab, which is just down by where we're having our morning teas and lunches. He's worked in the Industry 4 test lab for several years using a suite of industrial 3D printers, including resin, filament, metal and fibre reinforced printers. In addition, there are 3D scanners, desktop CNC and mechanical testing equipment. Outside of the test lab, he recycles 3D printed plastics and creates his own filament. So Daniel hasn't put all gushing things about himself here, so I'm simply going to say that he's a true expert in this area. He has a lot of knowledge, so again, tap into what he understands, ask him about the carbon printing, the metal printing and other things in the breaks, and make the most of being able to meet Daniel in person this week. It's online? Yep, all yours, Daniel. Let's make him feel welcome.
All right. Hi, everyone. So I thought I'd do a bit of a different presentation. Most people know about 3D printing and basics about what it does. So I thought I'd teach you about something a little bit different, about the background of how 3D printing started and how we got to now. So the history of 3D printing is actually older than most people actually think. So I'll start off with a question. Does anyone know when the first 3D printer was created? I guess at the year? Bit older than that. I'll give you some clues. It was the year the first game of paintball was played in the USA. Donkey Kong was released in the same year. It was the premiere of the TV show Dynasty in America. NASA launched the Space Shuttle Columbia. A Country Practice debuted on Channel 7 here in Australia. And it was the wedding of Prince Charles and Lady Diana. Anyone have... Close. 1981 was the first time that someone actually thought about 3D printing as a... So the first concept was actually thought of in 1981. This is the first sort of written down, someone thought about doing it. And it was created by a guy called Dr. Haido Kadoma. He was from the Nagoya Municipal Industrial Research Institute in Japan. He was the first to describe the layer-by-layer approach of additive manufacturing, and his idea was using a photosensitive resin cured by a UV light or laser in that sort of way. Unfortunately, he didn't actually go to the extent of creating a patent, so he's not commonly known as the father of 3D printing like the next person I'll speak about. But he was the first to have it written down, the idea of 3D printing. We next move on to 1986, and a guy called Chuck Hall, a guy from America. He was the first to actually have a patent on 3D printing, and he thought of the idea of SLA 3D printing, which is stereolithography, and that also used a photosensitive type of resin, which is cured by a laser as it goes along. So he had a bit of an interesting history. He actually worked at a furniture manufacturer as an engineer, and they applied resins, coatings on top of their furniture parts, which were all UV curing. He thought, oh, maybe I could use that to actually make 3D parts instead. So thankfully his company was actually quite progressive, and he actually gave him a workshop to develop the idea on his own, and he eventually created the idea of SLA 3D printing. So using very thin layers of resin, building it up over time to create the whole 3D part. So he went on to patent the idea, and then later created the company called 3D Systems, which is one of the largest industrial 3D printing companies out there today. So he's still actually involved in the company right up to now. And how SLA works is basically starting with a build platform and a tank of UV curable resin, and then a laser will shine up from underneath with some sort of optics to move it around. And that gets cured layer by layer, and then the platform slowly pulls out of the tank as it cures. So typically these parts come out very smooth and very fine details. The layer resolution can get down to about 50, 100 microns, so you get very smooth surfaces, and you often need some sort of supporting structures to hold it together, especially some sort of raft layer generally to hold it on the platform itself, and these get all removed afterwards. So we next move on to 1988. This was the first commercial SLA printer that Chuck Hall invented and made it available to the public. So this was known as the SL1, and you can see the very dated computer and the technology that he was using back in those days. In that same year, another form of 3D printing was also developed called SLS, which is selective laser sintering. And this was created by a guy called Carl Deckard from the University of Texas. He filed the first patent for SLS in this year. Interesting, he actually came up with the idea as an undergraduate student, engineering student, and he went on to complete a master's and PhD in the SLS 3D printing process. So this works slightly differently. It has a tank of basically powdered plastic, which will get laid on in very thin layers by a roller, and then that gets hardened and sintered together via laser, generally from above, and then the tank will slowly lower with the parts cured inside a cake of powder, as it's generally called. So the parts out of this are generally have a sort of rougher surface texture, but the main benefit of this is you generally don't need any sort of supports inside because the powder itself is supporting the part as it goes along. So you can get away with some very complex parts which don't need very much cleanup afterwards, other than removing any residual powder which is on the surface. We next move on to 1989, and the first patent for fused deposition modelling, FDM 3D printing. This is probably your most typical sort of 3D printing that you think about, a filament of plastic. This was created by Scott Crump, also from the US. He actually came up with the idea because he wanted to make a toy frog for his daughter, and played around mixing waxes and plastics in a hot glue gun to try to do this. And that eventually morphed into creating the patent for the FDM technology. He also created the filament to begin with as well, as this wasn't a standard thing. And in those days, ABS plastic was the most common sort of filament when they first started 3D printing. So he actually patented the idea with his wife as well. And the process works by having a spool of filament of plastic that's fed into a heated nozzle, which heats up the plastic and then extrudes it out layer by layer to create the 3D printed parts. And the build platform either drops down as it goes or the heads move up as it's printing. So this process you can obviously see the layers a lot more in this process because the layers are generally a lot bigger, around 100 to 300 microns. So it's a lot quicker than other processes, and you don't have to deal with any sort of fine powder or resins like the previous two technologies. So it's a lot safer and easier to use. You don't in that sort of industrial environment. So we next move on to 1992. So this was the first FDM printer that Scott Crump created, and he went on to found the company Stratasys with his wife as well. So Stratasys, if you've been to my lab, you'll see most of our machines are Stratasys. They are equal in size basically to 3D Systems as two of the big industrial 3D printing companies at the moment. So 1995, a different sort of technology was created, very similar to SLS. This is selective laser melting or direct metal laser sintering. So this uses a powdered metal instead of a powdered plastic. The main difference in this is the power of the laser that's required, and you often need more support structures with this technology compared to the SLS, as the powder isn't self-supporting in most cases. So basically works the same by a laser rolling over a thin layer of powder and then building up as it goes along. This was founded by the Fraunhofer Institute of Laser Technology in Germany, and other companies were also developing it at the time. EOS and 3D Systems were also looking at similar sort of technologies. So the parts come out of this printer are generally solid metal solidified via the laser, and titanium is one of the most common ones. Anyone who went to IPAS last night would have seen their metal 3D printers. They're all DSLM 3D printers. Has a generally very fine accuracy because the laser can be very small, and the surface generally is relatively smooth but has some texture to it as well from the fine powder. In 1997, this is going to be Billy's type of 3D printer, which he used in his micro lab. This is 2-photon polymerization, was developed by some people in Osaka University in Japan as well. So 2PP 3D printing is a little bit out there because it's on the boundary of physics. It's basically, instead of having a single light beam, it uses optics inside to create a single dot in midair, which is 2 photons in size basically, and then it uses that to move around to create very, very small objects. So they use a series of microscope optics, which it shines through, and that determines the size of the dot and the size of the parts that you can make. So Billy actually sent me some photos of some parts that he's made. So that's a standard pen in a tray. One of those dots is a 3D printed part. The other one is a speck of dust. So they are very, very small, and he loses lots of parts because he can't see them half the time. So that's what he was making, a very, very small Millennium Falcon. You can see the incredible detail on that. The picture is a little bit blurry, but it has very good detail and very, very tiny features. So this has lots of uses in the medical sort of industry when you're working with cells or sperm and eggs, which are at that sort of scale. So they can make things which can encase and move around objects in that sort of size. In 1998, the PolyJet technology was developed. This was created in Israel by a company called Object Geometries. And this works with a liquid resin again, but instead of having a tank of resin, it drops it out pretty much the same as your paper printer does at home. A series of small nozzles, and it drops out very small micro droplets. And then because it's a photosensitive chemical, it goes from a liquid into a solid, being cured by UV light as it's going during the print. So this means you can do fancy multicolour parts if you have multiple different heads on the 3D printer, and you can mix the colours however you like and have a supporting structure which can be removed afterwards. I do have samples here as well. I should probably show them. So you can 3D print parts in full colour and mix them all together however you like. This scan was actually of a full-size surfboard, which we 3D scanned and then printed in the full colour. So that captures all the detail and all the colour, and the scanner and the printer work together to create parts very detailed like that. So that's the basics of what the inside of the printer looks like. So the head on the left-hand side moves around and then builds parts on that tray, and that drops down while it's printing. So these parts are generally quite smooth because the layer heights are between 14 and 28 microns, so very, very fine, and you get a glossy surface finish because of that resin that it's been using. Then we jump on to 2004. So Dr. Adrian Bowen, he was a senior lecturer in mechanical engineering at the University of Bath in the UK. He founded a project called the RepRap project. This was an open source project aiming to create 3D printers which could 3D print themselves. So they were very simplistic and they used very rudimentary parts to be able to do that. And his idea was to make it cheap enough that anyone could be able to be 3D printing. He released the first RepRap printer in 2007 using very simple parts and very basic geometries. And being the nerds and scientists that they were, because they were recreating each other, he named the first printers Darwin, Mendel and Huxley, all after the evolutionary biologists. So this project then went on to develop in further years, and in 2009 was a big breakthrough for the field of 3D printing because the patents for FGM actually expired. So that is why in the mid-2000s 3D printing became a lot bigger, because the initial patents expired and then anyone could produce their own 3D printers. So this allowed other companies to create printers using the FGM process. However, Stratasys still owns the patent for the term FDM, so technically all other printers nowadays are FFF, fused filament fabrication, but it's more of a technicality more than anything else. So MakerBot was one of the first companies to actually create this type of FFF 3D printer, and they were very rudimentary at the start as well. Most of their printers were actually made out of plywood, and very basic laser cut parts and simple parts again, and using very early electronics. So they could only print very small sizes, and you were very limited in the sort of materials that you could use, and generally ABS once again, because that was the first which was created. And that's sort of what a modern FDM printer looks like nowadays. It's a lot simpler, you can print a lot larger, and the electronics are a lot smaller. So it's developed a fair bit in the last few years. In 2013 was another big breakthrough for 3D printing. It became a bit more public knowledge because President Barack Obama actually mentioned it in one of his speeches. And that actually made it a public term known as 3D printing, and it became public knowledge then. That same year, Markforged, which is another company for 3D printers, they were founded. And they developed a sort of a spur-off type of FFF 3D printing using a continuous carbon fibre. So that basically has two nozzles within the 3D printer, and the second nozzle lays down a continuous loop of carbon fibre within the parts that it's printing. So this can either be, it was started with carbon fibre, but it can now be fiberglass or Kevlar as well. And you can see this part, the orange part is the Kevlar, and the black is the plastic itself. So it lays them down layer by layer, and that increases the strength and the stiffness of the parts by having those continuous fibres within the print. So that's what it looks like inside the printer. You see the loop of Kevlar, and the black is the 3D printed material. In 2015, a different sort of 3D printer was created called CLIP. This was developed by Carbon 3D, and it stands for Continuous Liquid Interface Production. So it's a form of SLA 3D printing using a laser or a projector in this sense, and shines it up from underneath with a vat of resin and then pulls it out as it goes. There's some fancy technology and chemistry in there that they have a oxygen permeable window in the bottom and a dead zone, which means they can print a lot faster as they don't have to lift the part and drop it back down into the resin for each layer. So it means they can print much quicker than typical SLA 3D printers. Interesting enough, Stratasys actually now own Carbon 3D. They now also own Object 3D. And they were looking to actually own the next company as well. So in 2017, Desktop Metal was created. They use a bound metal deposition type of 3D printing. So this is different to the laser-based metal 3D printer. Instead, it uses a feedstock of material, basically a rod of powdered metal with plastic and wax inside. It then prints it as quite a thick paste, and then has to debind and sinter the parts to remove the plastic and waxes, so you're just left with the solid metal at the end. This is more of an office-friendlier version compared to direct metal laser sintering, as you don't need that powdered metal. It's a bit safer to handle, and it's the only downside is it takes a bit longer and you have to do a bit more processing steps in there. So that's what the printers look like, the 3 steps, the printing, debinding and the furnacing. So nowadays there are 18 plus different types, technologies of 3D printing, and I'm sure there is more than that. They're very niche fields, especially in some of the biomedical applications. They create their own printers for custom tasks, and it's estimated it's worth $23 billion US, the market value for 3D printing. So this slide's a bit of an old picture of the different types of 3D printers which are out there. But you'll see there's the vat polymerization type, which is the SLA, and then the continuous light processing. So they all use a laser or a projector with a vat of resin. FDM uses the extruded base, generally plastics or composites. The material jetting, which is what polyjet comes under, some sort of droplets of material going in. There's also binder jetting, which uses powdered plastic or metal with another droplet of a binder in there. Then there's multi-jet fusion, and then all the plastic fused together by lasers and metals. And then there's also direct energy deposition, which uses electron beams or lasers. And there's another one which I haven't really heard much about, but apparently uses layers of paper and stacks them up to create a 3D object. But they're obviously a bit less common because the structural integrity probably wouldn't be there compared to other materials. So the main advantages of 3D printing is you can rapidly prototype. You can make parts within the same day. You don't have to outsource them, and the printers themselves are quite cheap, so they can be housed in most locations. You can have a digital inventory, so if your part breaks on your machine, you can keep a track of all those parts, and you can just reprint the part that's broken. This is very good for remote places where they can't ship in parts in time. There's actually 3D printers on lots of Navy ships at the moment. Also on the International Space Station has a 3D printer, so they can recreate parts if they need them. On-demand production, you can only have to 3D print what you need. You don't have to wait or create any sort of tooling or injection molds like traditional manufacturing. There's a wide range of materials, plastics, metals, composites. The minimal material, only using what you need within the print. You can create very complex designs, as you can get away with internal channels and cavities, and you can put supports only where they're required. You can customize the parts. Each 3D print, you can make them different to whatever you want. You can put labels or logos on. You can make them to suit different people in terms of prosthetics and things like that. And then there's the flexibility that you don't always have to print. You can do it in lots of different locations, and it's fairly easy to use nowadays. Some big use cases in the last few years which are a bit abstract. So you can 3D print homes now out of concrete, which use the same sort of technologies. You can 3D print implants and prosthetics for medical applications. You can print models and architecture models or scale models of everything that you like before you go into producing the large object. And there's the 3D printer on the ISS as well, as I mentioned just before. So that's about it for my talk. Any questions?
Excellent. Thank you, Daniel. Questions for Daniel? You can see that there's some knowledge in this mind that you can tap into right now about 3D printing. It's extensive. Any questions? Go on, Mark.
What's your favourite 3D printer and why?
My favourite 3D printer? Probably the full colour resin printer is pretty cool because you can mix any colours that you want, and you can recreate parts that look just like the real thing. And you can get basically injection molded quality straight off the printer.
Go ahead. No, this is one of the ANFF people who asked him up.
Go ahead. Thank you for your presentation. What's the manufacturer of the 3D printer in our laboratory?
So we have a mix of a couple of different manufacturers. We have Stratasys, we have MakerBot, we have Bamboo Labs, we have Desktop Metal, as I mentioned. We have Lulzbot, we have Markforged, and Formlabs. So we have a range of different printers. Most of the companies only specialise in one or two types of printers, so you have to get them from a number of different companies.
Okay. So,
We have lots of materials. We probably have around 50 different materials that we can put into the printers, plastics, metals, composites, reinforced materials, carbon reinforced materials, high temperature, high grade materials. So it depends on the application. We can probably get in a material which suits it.
Up here.
Yes, yep. Yeah, so there's a number of materials which have high durability and like impact resistance, but it's only on a certain number of printers, so not every material will be suited to that sort of application. So we can print things like nylon 12, polycarbonates, Ultim and Peak type materials, and they're very high wear resistance for an end-use application.
I will come down here next.
What's sort of the future applications that they're sort of looking into that you might be able to?
It's pretty advanced now. You could pretty much print anything that you want. In modern times, the 3D printers have become a lot easier to use. They do lots of internal tests and checks, which you had to do manually before. So generally just 3D printing getting smarter. I could see things like artificial intelligence coming in to set up prints for you. That could also happen in the CNC world, setting up their own programs so you don't have to be involved as much. Yeah, just making them smarter and easier to use. There's a wide range of materials now, but they're constantly bringing out more. So maybe more specific materials for niche applications as well. So most of the polyjet resins are basically an acrylic, so they're quite brittle generally, especially the full colour parts. There is a couple of other resins which we can load in which are more tough and reinforced, but you don't have the selection of the colours then as well, so there's a bit of a compromise between the two.
So we have a question online. Looking back, what would be the single piece of advice you'd give to somebody who's just starting out with 3D printing?
Well, 3D printers are so cheap nowadays that anyone could really go buy one. But I think it's important to know that I see lots of people trying to sell 3D printers. They buy it for a project, they print some trinkets, and then they want to get rid of it. Because it's very cool technology, but you don't always need it all the time. So there is a lot of other maker spaces and service bureaus out there which can do the printing for you, especially if you only need it for a short term of time. So the test lab, for example, we can print parts if you just want one or two off and you don't want to invest in the technology yourself. But I think it's important to have a project in mind when you want to print something, and figure out what can and can't be printed, and what sort of the limitations are. So one of my first things that I 3D printed was in high school. I printed an action figure, basically recreated an action man, fully 3D printed. And that was a cool project because every single part was 3D printed. And then I could figure out what polishing and painting techniques you could do in it as well. So it's quite versatile in what you can make and develop with it.
Yes.
Have you had any experience working with 3D printing technologies that can also be used to repair existing parts in UniSA in the future?
Yes, there's a big industry in remanufacturing and repair. The 3D scanning is probably the key technology in that terms. Really complex industrial parts, if you can 3D scan them and create the CAD model, the CAD model is the most important thing you need to be able to remanufacture it. So you can then 3D print the parts that you need, any sort of spare parts. As I mentioned, International Space Station, Navy ships, they're doing a lot of that. They're producing their own parts, which have broken while they're on their missions. So it's a big industry to recreate 3D printed parts.
Just jumping on what you said before, are there any sort of big limitations on what you can't do?
Depends on the technology. Each technology has its limitations in terms of strength or quality, or the size has limitations. Most printers are generally about the size of a loaf of bread is what you can print. If you want to get any bigger than that, then you become into limitations with warping and shrinkage. If they become too big, then the parts will start to shrink and cool too quickly. So there is limitations on the size and the sort of materials that you can use. And then you get more expensive as you go into those industrial grades of materials as well.
So Daniel, we've got another question online about bioprinting, and what about printing meat and other products like that? Can you say something about bioprinting?
It's not really my speciality. I haven't done much bioprinting, but I know there's lots of research and development in there, mainly in biology labs and things like that. They can recreate skin grafts and scaffolds to recreate broken limbs. I know there was a project they were working on an ear. They were 3D printing the scaffold of an ear and then grafting skin on top of it. So there's lots of applications for surgical training models. There's a company in Adelaide which does training models all 3D printed that surgeons can train on before they go into the surgery. And lots of hospitals nowadays are developing or buying their own 3D printers so they can do that sort of training models to prepare for their operations as they go.
I guess one more question this time from me. The transition, when 3D printing was first out there, people were using it for prototyping, but you've hinted on this already that the prototyping's still done, but people are actually making products now, finished products with 3D printing in some cases, and I'm thinking of a space application as well as some other things. Can you comment on that?
Yeah, so to start with, and sort of a underlying theme at the moment is people think 3D printing is only for trinkets and toys and things like that. But lots of companies now 3D print their own components in their assemblies of parts. Lots of manufacturers in the automotive industry create 3D printed parts. In the aerospace industry, create 3D printed parts. The challenges in some of those areas is getting the material certified so they can actually use it. And there's a big push at the moment to get certifying the actual technology itself, so it can be unknown what sort of standards it can live up to, what sort of strength it has. I think the aerospace is probably the next big area that it's going to push into, having end-use parts, especially in metals. That's probably one of the bigger area, and the engineering-grade polymers.
And is it true, Daniel, that something we've printed at the test lab is currently in space?
Oh yes, yeah. We printed a part for a satellite out of an ultram-grade material, which is a low outgassing in a vacuum, and that's on a satellite at the moment. We've also used that material for vacuum chamber testings as well, so they test their satellites in a vacuum chamber, but if they have materials which in there which outgas while it's in a vacuum, they won't get very good results. So we print lots of jigs and fixtures for them to hold their satellites in the vacuum chambers while they do their testing.
Excellent. All right. In a moment, we'll thank Daniel. I can't remember who won the prize. So Daniel's going to hand deliver it to you while we cheer like crazy because he did such a great job. Thanks very much again, Daniel, for your excellent talk and bringing your knowledge to us all. Hopefully you enjoyed that journey through 3D printing. It's lunchtime. Belle has given me instructions to let you know that they're not boxed lunches today, it's a shared lunch. So there's a lot of us, so she's just asked you to just think about each other as you don't take a whole platter for yourself of the most delicious thing, but just think about each other as you take